Hello, my name is Oliver Martin and welcome to my model/presentation surrounding the processes of polypeptide synthesis. To commence, polypeptide synthesis is considered one of the most significant hereditary processes within the human body. The terms polypeptide and synthesis essentially refer to the creation of a polypeptide chain. These chains are so significant as they further go on to create proteins that function in the roles such as transport, structure, protection, and even in some cases, the creation of enzymes. This process of creating a polypeptide chain can be broken down into two major steps, transcription and translation, which involve the use of a DNA, a codon and anticodon sequence, enzymes, as well as messenger, ribosomal, and transfer ribonucleic acids. DNA, found within the nucleus, is built up of structural components known as nucleotides, consisting of a phosphate molecule, deoxyribose sugar, as well as a nitrogenous base. There are four types of nitrogenous bases, which include adenine, thymine, guanine, and cytosine. Adenine and thymine bind together as they only use two hydrogen bonds, whereas guanine and cytosine bind together because they use three hydrogen bonds. Transcription, the first step of polypeptide synthesis, is essentially the process of creating an RNA copy of a gene's DNA sequence. Initially, the DNA's double helix structure is split into two separate strands through the action of unzipping by the DNA helicase enzyme. The enzyme of RNA polymerase then subsequently synthesizes a new mRNA strand based on a templated strand. However, mRNA replaces thymine with uracil so the mRNA sequence can undergo translation later in the ribosome as the thymine is incapable of leaving the nucleus. Once the newly copied mRNA sequence is complete, it leaves the nucleus via nuclear pores to which it enters the cytoplasm. Following the mRNA's exit from the nucleus, the two strands of DNA recoil back together with the creation of hydrogen bonds between the nitrogenous bases. From there, the newly copied mRNA sequence makes its way to the rough endoplasmic reticulum for the process of translation. Translation is the second major step of polypeptide synthesis and is classified as the process of converting an mRNA sequence into a sequence of amino acids known as a polypeptide chain. Once the mRNA molecule enters a ribosome within the endoplasmic reticulum, ribosomal ribonucleic acids will then attach and stabilize the mRNA in order to facilitate the subsequent binding of transfer ribonucleic acids. Ribosomal ribonucleic acids perform this action through the utilization of its two subunits, the small subunit and the large subunit. Once completed, tRNA molecules then signal for specific amino acids that are complementary to the mRNA sequence, to which the tRNAs then attach their anticodon sequences with the mRNA codon sequences. Codons are sequences of three nucleotides along an mRNA sequence that code for a specific amino acid or a stop to the process, as seen in the table, whereas an anticodon is a trinucleotide sequence found on the end of a tRNA molecule that are made to correspond with a codon sequence to deliver a specific amino acid. As the tRNA molecules progressively deliver amino acids, the tRNA bases detach and disperse, whereas the amino acids bind together using peptide bonds, which then results in the product of a polypeptide chain. Once a polypeptide chain has been synthesized, the process of transforming this linear sequence of amino acids into a protein begins. There are four structures part of making a protein. This includes primary, secondary, tertiary, and quaternary. The newly synthesized polypeptide chain is considered the primary structure, to which the secondary structure occurs when the peptide backbone of the chain undergoes hydrogen bonding, causing the amino acids to fold in a repeating pattern, either in the form of alpha helices or pleated beta sheets. This structure then subsequently turns into a tertiary structure through the overall three-dimensional folding of the chain and unification of secondary structures, before finally entering a quaternary structure where multiple polypeptide domains are arranged intricately to form a protein. Once again, thank you for watching my model/presentation surrounding the processes of polypeptide synthesis. I hope you found this helpful.